Hello students, in today's class you will learn about different preventive measures for diseases. First is public hygiene and the second is healthy environment. Now see about public hygiene. Public hygiene means cleanliness of the public places. In this, public places involves proper disposal of human excreta and domestic waste. The sewage and chemical waste should be disposed properly in water bodies, and before disposing that waste, it should be treated first. The sewer lines should be properly connected to sewage treatment plants. All these comes under public hygiene. Now the next is healthy environment. To maintain healthy environment, we should prevent the spreading of diseases due to the breeding of mosquitoes, houseflies and microorganisms. So, stagnant water is the best breeding place for mosquitoes. So, we should take care that there should be no stagnancy of water in our surrounding, because that provides a good breeding medium for the mosquitoes. Garbage should be kept in covered bins so that flies cannot breed on them. Contamination of drinking water should be avoided, because contaminated water is contaminated when it is mixed with the fecal matter, means the solid waste matter of human as well as animals, and it causes a number of diseases. The next preventive measures, that is, it also comes under the prevention, that is vaccination. Vaccination, the word vaccination has been taken from a Latin word, that is vacca. V-A-C-C-A. Vacca means cow. And this vaccination, it's not a process. This is a practice. This word vaccination, it's first time conceived by Edward Jenner when he observed that the milkmaids in his area, they are not suffering from smallpox as other persons, they are suffering from smallpox. As the milkmaids, they were in contact with cows, they were suffering from cowpox, and the person and the people, those who were not in contact with cows, they were suffering from smallpox. So first time he taken the pustules of the cowpox through an injection and injected that cow pustule, means pus, that injected to a boy's body, that is 8 years old boy. He got the material taken from the cowpox pustule and introduced into the boy's body. The boy's body developed pustules, which later on formed a scab and healed leaving only a scar. Means after injecting that cowpox pustule, the boy's smallpox is being cured. So further he tested his idea and injected the boy with smallpox virus after 2 months again, and then he observed that there were no sign of smallpox on the boy's body. In this way, Jenner vaccination experiment was successful, and later Jenner's finding was confirmed by Louis Pasteur, and he developed vaccine for other diseases like anthrax, chickenpox, cholera, etc. So, the vaccination, it's a practice of artificially introducing germs or germ substances into the body for developing resistance to particular diseases. I hope the definition of vaccination is clear. It's a practice of artificially introducing germs or germ substances into the body for developing resistance to particular diseases, as Edward Jenner done with the case of smallpox. Now there are different methods of preparation for preparation of vaccine. This vaccine, usually in this vaccine, a germ means the disease causing germ or any substance of extracted from the germ body is directly introduced in the body, either orally or through injections. As you know that orally polio drops is being given to small children, and as an injection so many vaccines are there. For an example, TAG vaccine is there. When these germs injected into the body of a human being, the WBCs are get stimulated, means they are being induced to WBCs become active and they produce antibodies against these germs. So in this way, the person's body develops resistance, means capacity to fight against that particular disease is being developed in that body. Now the different methods are, first method is using killed germs. In this method, the germs, means the disease causing germs, are used or injected in killed condition, means they are completely in killed, dead condition, so that they cannot cause any harm or cannot cause any type of disease. In the second method, using living weakened germs. In this method, the germs are in living condition but in weakened condition. Weakened means they are treated in such a way that they have become very weak and they cannot cause the disease, and they can induce antibody formation in the body. For an example, measles vaccine, tuberculosis vaccine, that is BCG vaccine for tuberculosis, these are prepared by using this method. Now the third method is using living fully virulent germs. Means in this case, the vaccine is, through this method, smallpox vaccine is prepared. In this case, a person is inoculated with cowpox virus. Inoculated means injected. Means cowpox virus is very much similar to smallpox virus. So that cowpox virus in fully living condition is injected into the body so that WBCs get confused with smallpox virus and they will produce antibodies against smallpox. Now the last method is using toxoids. Toxoids are actually the toxins. Toxins means the chemicals which are being produced by the germs when they enter in a person's body. That toxins secreted by any kind of germs, for an example bacteria, and that poisons are made harmless. Means when that organism will secrete toxin that is extracted from that person's body and it is treated with formalin solution to retain the capacity to produce antibodies. Means when it is treated with formalin, its toxicity is being reduced, but it can induce WBCs to produce antibodies. This is the sum up of vaccination. For more detail, just go through your textbook. Today's homework, question number 1, write about the preventive measures for our good health. Question number 2, what do you understand by vaccination? Question number 3, explain different methods which can be used to prepare vaccines. Write these answers in your biology notebook and keep a record of these questions of everyday class questions shared in videos. Thank you.